All right, I'd like to officially welcome all of you to the California Initiative to Advance Precision Medicine Symposium on Cancer Disparities Research. I'm Julianne McCall. I'm very proud to be the director of the initiative and serve as the director of precision medicine within the California Governor's Office of Planning and Research. I'm joined by my extraordinary staff, including Dr. David Reiner, who will be emceeing the remainder of this symposium. Before I pass off the microphone, I just want to thank the teams for the tremendous dedication that you've all shown. I remember May 1st, 2019, we gathered in the Governor's Office conference room to launch these projects, and the energy of the conversation and just the extraordinary hope shared across teams just gave me, enormous inspiration. And I'm very proud that we've made it to this stage, very proud as a state to have supported such world-class researchers and community partners trying to help change and update the paradigm of community-driven and patient-focused research in cancer. So with those very brief remarks, I'm going to pass it off now to Dr. David Reiner, Science Officer in the Governor's Office of Planning and Research.
Hi, everyone. It's a pleasure to be here with you. Thank you, Dr. McCall, for that introduction. My name is David Reiner. I'm a science officer, which means I oversee our demonstration research projects, including our 3 projects on cancer. And we're delighted to welcome you to this symposium on those projects. We have 3 presentations today, one on each of those 3 cancer projects. And I'll just get right into it. Excuse me, one more thing. After the presentations, we'll have a discussion, followed by kind of a closeout of different closing remarks. So with that, I'll introduce our first speaker, Dr. Manali Patel. Dr. Patel is an associate professor in the Division of Oncology at the Stanford University School of Medicine. Her research involves developing and implementing interventions that overcome systems-level barriers to improve equitable, value-based cancer care delivery. Dr. Patel received her MPH and MD from the University of North Carolina and completed her residency in internal medicine, fellowship in hematology and oncology, and master's in science and health services research, all at Stanford University. Dr. Patel is joined by a few members of her team today, Dr. Lisa Goldman-Rosas, Isabel Duran, and Emily Wood. In this specific case, Dr. Patel had a family emergency and has pre-recorded her talk. So I'll stop sharing the screen and Emily Wood will share the screen with that pre-recording.
Thank you, everyone. And just a note that we've noticed the audio is a little bit low, so you may need to turn up your speakers as I share it. I will try and have mine sharing at top volume. Thank you. I'll get started.
Hi, I'm Manali Patel. I am an oncologist at Stanford. I'm very happy to present our work. Again, we can't thank the funders and sponsors, CIAPM, enough for taking a chance on our project and allowing us to successfully, we think, address Latinx cancer care equity. And hopefully, with the preliminary results we present today, you will also agree that we've successfully achieved our aims. I'm so sorry I can't be there in person. I had a family emergency. We have an incredibly amazing, dedicated team of partners that include patient advocates, including our co-PIs, Isabel Duran, Lisa Goldman-Rosas, who's an epidemiologist at Stanford, Dale O'Brien, who's a community-based organization, who's Cancer Patients Alliance, Zach Kuntz, who's a medical oncologist at our local partner, Pacific Cancer Care in Monterey County, Dr. Naquacha and Dr. Cuevas, who represent the federally qualified health clinic in Monterey County, Salinas, and our wonderful team of researchers, including Emily Wood, Lanzao, who's our statistician, and Ms. Reynaga, who's our research assistant. We also are very fortunate to have an incredible team of community health workers who are located in Monterey County, two of whom are working in Pacific Cancer Care, Jerry and Leslie, and Miriam and Cassandra, who are working both in La Clinica. Our project aims were quite ambitious, but I'm happy to report that we have completed 3 of the 4 and are in process of assessing the 4th. Our first aim was to establish a community advisory board that was focused on addressing cancer care disparities with a keen focus on precision cancer care, and then to develop a community health worker precision care intervention in Pacific Cancer Care, a multi-level intervention in collaboration with our community advisory board to overcome disparities in equitable access and receipt of evidence-based precision medicine cancer care treatments, as well as testing. And then our third aim was to implement and evaluate this intervention, and finally to assess the financial impact of the intervention. Our timeline is shown here. We began in 2019, prior to the pandemic. We had our first CAB meeting held in August, which was in person, and now we have subsequently, since then, launched not only the multi-level intervention and the randomized trial with our first patient that was enrolled in 2021, but we also began a wraparound service with our federally qualified health clinics to also conduct an aspect for screening and prevention to evaluate for genetic testing for individuals that may have a very high risk of family history or may qualify for genetic testing. Our plans are to complete these projects by next year, as I mentioned, with AIM 4. We've also added on several additional projects, including obtaining funding for navigation services through the American Cancer Society, as well as several innovation grants to expand work, specifically as it relates to health-related social needs screening for patients that are enrolled in the current trial, as well as additional patients that are being seen at Pacific Cancer Care. Our AIM 1 shows here our Community Advisory Board. Again, as I mentioned, in August of 2019, we held our first in-person Community Advisory Board, and subsequently, due to the pandemic, had to quickly shift to virtual meetings, which has been the preference, actually, for most of the advisory board members. We have our last advisory board meeting next week, which is really exciting in Monterey County, and are excited to present these results there, as well as discuss sustainability. In between our meetings, we also were very fortunate to have Isabel Duran's group create a series of essentially newsletters to be disseminated out to all of the community advisory board members, and many of which were either discussing reaching the goals or highlighting community health workers or actually talking about getting out the vaccine, given the fact that we quickly had to shift gears as the pandemic encroached upon us. I'm going to talk briefly about the prevention and screening, which was not actually the cancer care delivery intervention, but it was an add-on. Here we were really focused on trying to do what we know is already demonstrated as part of good quality care for individuals, and it's really trying to prevent cancers before they occur. But as part of this, we also wanted to include precision genetic testing for individuals. And so our team, along with the community advisory board members, adapted the Stello tool, which is a screener for high-risk individuals that may have family history. It actually spits out a score that's validated, and for individuals who score greater than 1, greater than or equal to 1, there's a triage and they are actually recommended for genetic testing. We're happy to share that with people if they're interested. Our preliminary findings were we conducted several outreach interventions and several outreach events, and were able to distribute almost 3,500 flyers. We also had 450 community health worker contacts with individuals in the community regarding basic screening. 158 people were approached to complete the Stello Family History Screener, and of those, a pretty high success rate, 153 people completed it due to our wonderful community health workers. 33 or 21% had a Stello score of equal to or greater than 1, which was indicating that there was a potential benefit for genetic testing. 7 of those ended up receiving genetic testing through Invitae, and received post-test counseling, which we created a pretty impressive workflow for with Clinica. I'm going to focus now on our randomized trial, which is what we were really funded for, and this was to determine whether a multi-level intervention that we adapted could improve patient knowledge of precision cancer care. That was actually an outcome determined by our community advisory board. Receipt of evidence-based treatment, specifically looking at molecular and genomic testing, but then also the subsequent treatments that would ensue after testing. Looking at patient activation and quality of life, mostly as an exploratory outcome, among low-income and racial and ethnic minorities who were diagnosed with cancers, we had a stratified enrollment looking at individuals for whom those genomic and molecular tests were recommended. And our hopes were that we were to be able to improve these outcomes for individuals in the intervention more than usual here. Our intervention, as I mentioned, was multi-level. We started with the clinician-facing intervention, which included clinician training after our ASCO annual meeting. Dr. Kuntz and several others in Pacific Cancer Care would conduct biannual trainings for clinicians to ensure that they were up to date on what the molecular marker testing should be for specific cancers, and ensuring that there was a pathway before we started. We ensured that there was a particular pathway along with our payers for molecular and genomic testing to be obtained on samples. Initially, our thought was to send the samples to Stanford, but we created a collaboration with Foundation One. Our second aspect of the intervention included community health workers, and we heard from many of our community advisory board members before we started that patient education regarding tumor testing was really important. It would add this extra layer of activating patients such that they could hopefully encourage their clinicians to order this test before they would start with therapy, and then also to help others in the community who may be diagnosed. And so we created a series with a now resident at UCSF, she's currently a medical student in Chicago, created these wonderful infographics. We created a series of patient-facing informational tools, and then one-to-one structured conversations with our community health workers. In addition, we were able to receive follow-on funding from ASCO of a mentee of mine, Gladys Rodriguez, who's now at Northwestern, who created these wonderful patient-facing videos in Spanish that further augment the education that was being provided by the community health workers. Our inclusion criteria was pretty open. We wanted to include anyone with any cancer diagnosis. Again, I mentioned we stratified the analysis, or stratified the enrollment by those cancers that would require genomic and molecular testing per NCCN guidelines at the time that we started the trial in 2019. We included individuals that were 18 years of age or older, who had a capacity to verbally consent in either English or Spanish, and who had either public insurance, low income status, or insurance that was provided by a local health agricultural company in the area, or who were uninsured. And we excluded anyone who was planning on coming to Pacific Cancer Care only for a second opinion and were not planning on receiving additional care. You can see our study schema here. We randomized. Our goal was 110 to be able to see a benefit in patient knowledge that was clinically meaningful of about 1 point on a validated standardized questionnaire called the Molecular Profiling Knowledge Questions that we had adapted. We also obtained several repeated measures. So we not only obtained information about patients' quality of life and their satisfaction with decision, but then we also repeated those measures over time up to 12 months. The duration of interactions with the community health worker and the clinician-facing intervention was over the year. We can show our preliminary results, which are actually hot off the press. We assessed, this is our flow diagram and our study flow of all patients through our randomized trial. There were a total of 1,390 that were randomized or assessed for eligibility, We excluded quite a few, so these individuals were screened from the electronic health record, and many of these did not have a cancer diagnosis or were not eligible. There were 71 who declined participation. Of those, we've reached our target of 110. 53 were randomized to the control, and 57 were randomized to the intervention. There were several that had died after follow-up at 3 and at 6 months, and there were only a handful, 3 that had withdrawn in the control group, and 2 that withdrew in the intervention group. We analyzed all of these individuals through an intent to treat, so I apologize, this slide is incorrect. There was an analysis of 157 in the intervention group and 53 in the control group. Our preliminary demographics for the total cohort was a median age of 55 years. This is very different than most of my studies, very young population. Most indicated that they had a race of, actually did not list their race, and when we were able to obtain handwritten descriptions by race and ethnicity, most had indicated either Mexican or Hispanic as their race and did not preferred and did not think that their race was listed here. So that is the majority of the individuals. There were about 89 who had indicated and self-reported Hispanic as their ethnicity, with Spanish as their primary language. The majority had a high school diploma or GED or less, and the majority also had low annual incomes as indicated in the eligibility criteria. We also saw that a high proportion had other cancers, so this was a mix of both lung cancer as well as soft tissue cancer and leukemia. And then we also saw that individuals had a high history of GI cancers and breast cancers with a stage 3 or 4 diagnosis. Our primary outcome was looking at the change in patient knowledge over time from baseline to 3, 6, and 12 months. And this slide is showing at baseline the mean score. So you can see here in the blue dot, which is very small, so I apologize, but the blue dot shows the control group. And this is the mean score on a 6-item questionnaire that assesses whether or not individuals may understand the difference between somatic and germline testing, and also the impact of testing tumors in particular and the impact on treatment choices. And so you can see here they're pretty evenly matched at baseline, which is what we would hope. And then over time, the intervention group began to acquire more knowledge, with the split and difference appearing to be statistically significant over time. Again, we have not run statistics on this as of yet. We're waiting for our 12-month data to be completed. But this is actually what the scores are showing now, that we did achieve, it appears, our primary outcome of improving patient education over time. We are in the process of obtaining our secondary outcomes, which are evidence-based, receipt of evidence-based care, as well as looking at the impact on quality of life. And then we have conducted a series of implementation evaluation interviews to obtain more information about how to implement this at scale. One success of this project is that this is now part of usual care at Pacific Cancer Care. We've completed the study, and the community health workers are completely funded by the clinic itself. They have continued the intervention, as well as I mentioned, added on several other exciting interventions, and are really just part of usual care at the clinic, and continue to educate individuals not only on precision medicine, but are now educating individuals on advanced care planning, as well as conducting health-related social needs screening. And finally, we are hoping to develop a community toolkit that will have recorded trainings that are free online resources for community health workers nationally. We're hoping to include all of our materials that have been created by our group, and we'll be making that freely available online, and we'll be sharing that with you all. Again, I can't thank the sponsors enough for taking a chance on our study and allowing us to demonstrate the benefit of multi-level interventions on precision medicine cancer care, not only at the point of screening and testing, but also at the point of diagnosis and beyond. So we really appreciate it, and again, I'm looking forward to hearing about the questions.
Thank you, Emily, for playing that video. We have time for one question. I know Dr. Patel isn't here, but Emily and Isabel are here, so maybe they could answer the question on Dr. Patel's behalf. I see Dr. Ramos with your hand raised. Go ahead.
Yes, so it was, I think, a really good study. Can somebody just give a quick synopsis? It was very difficult to hear.
Yes, I'm so sorry about that. So our study had two parts. The main portion that the majority of the presentation was covering was our randomized controlled trial that took place in a community oncology setting, Pacific Cancer Care, where we had folks who were randomized either to the treatment group where they would receive some supplemental support, including education about precision medicine that was provided by a community health worker. We had 2 bilingual community health workers who provided that education in one-on-one sessions where they would talk about, what is tumor testing, how might it guide your care, and then in the other group was the control, so continuing with usual care. We did surveys at baseline, 3, 6, and 12 months, and the primary outcome was looking at change in knowledge related to precision medicine. And so we had a 6-item survey that looked specifically at knowledge about precision medicine. It was adapted from a previously used measure, and that was, at the end where you were seeing the graph displaying the change in knowledge scores over time. Very preliminarily, it is looking like our group that was working with community health workers had a sustained increase in knowledge that was separating over time from the control group.
And so, sorry, all participants were diagnosed with cancer. Is that correct?
Yes,
Okay.
all were diagnosed. Thank you.
And Dr. Ramos, from the community health worker piece where we were implementing them inside the clinic system with the intention, hopefully, of moving people into screening early for that early intervention and not late stage diagnosis. And of course, what we showed, in my mind, something that I've always been conscious of the value add of a community health worker is that they do outreach as well as inreach, so that they work not only with patients already there to help them get back to their screenings, et cetera, but it also reaches into community to raise that awareness and diminish some of the fear and other issues, including language barriers and some insurance barriers, so that we can move them into those screenings and into a medical home. And so that what we tried to show was the value add both already in the cancer care, but also external to the value add of a community health worker along the cancer continuum. And while we don't have as much data like they do on the patient care side, what we saw was a real increase in activity of the community health worker, but we also saw the response of community who were constantly calling back to those workers for assistance and helping move them into those screenings to get better education and also to get those extracurricular resources that you don't normally find and were even more important during the whole COVID era, in which there was a lot of challenge for this community. So I really think that the experiment for me, I think, worked very well. And I'm hoping that as we see more intentional commitment to having community health workers throughout systems, and not just in community, but working hand in hand and bridging with systems, I think we can begin to address some of those disparities.
Thank you, Emily and Isabel, and thank you for your question, Dr. Ramos. We're going to move on to the next talk, but we have about an hour of discussion at the end of the talks, so we can get back to some questions people had. So moving on to the next talk, this is from the University of California, San Diego, Doctors William Kim and Pablo Tamayo. And just to introduce them, Dr. William Kim is assistant professor in the Department of Genomics and Precision Medicine at UCSD School of Medicine, and his research focuses on understanding molecular mechanisms of tumor-promoting genes to guide improved therapeutic strategies. He received his PhD from Duke University in molecular genetics and completed postdoctoral fellowships at the Dana-Farber Cancer Institute and the Broad Institute of MIT and Harvard in medical oncology and cancer. Dr. Pablo Tamayo is a professor in the Department of Genomics and Precision Medicine at UCSD School of Medicine, and his research focuses on cancer pathways and targets, treatment outcomes, disease subtypes, and drug responses. He received his PhD in physics from Boston University and completed postdoctoral fellowships at Boston University, Thinking Machines Corporation, and Los Alamos National Laboratory in physics and computational science. They are joined today by members of their team, including Dr. Rebecca Shatsky, Dr. Barbara Parker, and Dr. Connie McDaniels. And with that, take it away, William and Pablo.
Okay, thank you. Could you hear me?
Yes.
Okay. So this is this picture is the way we envisioned this project at the beginning. Now that we are completing it, we did, I think, most of what we proposed and a little bit more. So I'll try to very quickly give you an overview. So we are a large team, and what I'm presenting is the result of the work of many, many people working together. Our system is called Celsius, and it's based on an early proponent of precision medicine 2,000 years ago in the times of the Roman Empire, Aulus Celsus, who advocated that medicine should be rational. We use a paradigm based on, it's a Bayesian information theoretic paradigm introduced by Alan Turing and I.J. Good in times of when they were cracking the Enigma machine during the war. It's not a black box, it's sort of AI-like, but this is sort of a more transparent approach to that. And it's rational, hopefully it's Pinker approved. Hopefully it allows to look at cancer in a different way. So the first aim was to make this model to characterize pathways, in particular the ones relevant to triple-negative breast cancer. And this is the way the system looks like today. It has everything that we proposed originally, and it has a few extra features that I will describe. It allows to match treatments with tumor samples using genomic features that could include mutations, pathways, protein profiles, etc. So it's a very general system. It can work on any cancer. It can be specialized for triple negative or even subsets. So it has great flexibility. One part of the system is the model building, which is shown on the left. Is basically training the model with existing response data. And this could be from drug screens, CRISPR screens, could be data from patients that were treated in the past, could be organoids, could be PDXs, could be any type of response data that can be used to train the model. And then on the right side, when you have a new sample, the model will try to classify it and tell you what is the likelihood of response to a collection of pharmaceutical interventions or therapies. And it will give you a nomogram showing why the model thinks that there will be response to each of the drugs. And it will also tell you which drugs are more likely, and also which drugs are likely to be counterproductive. So it shows you both sides. The model is built on a model of tumor states that we developed as part of another project sponsored by the National Cancer Institute. And then we basically map our cohort and we projected. So here you can see in this map of tumor states, our breast cancer samples tend to go into certain states more than others. So this is a very useful way to understand what is happening. So I'm going to give you a very brief example how the model predicts response to a drug, dabrafenib, which is used to treat, for example, BRAF mutant cancers like melanomas and others. So the model finds genomic features that correlate with the response. This data here is cell lines that either respond or not to the drug. And here it finds these genomic coordinates and states that are relevant to the prediction. Following that weight of evidence, it creates conditional probability models to describe that, and then combines them on a nomogram where you can see graphically the amount of evidence that each feature contributes either in favor of response or no response. Once that you do this for hundreds of drugs, according to whatever is the input data, you can sort them and see what are the drugs that have the highest likelihood to affect the tumor. And in this case, I'm showing you a melanoma cell line. And these are the model predictions. And every cross at the end of this bar is a case where the cell line is actually sensitive to the drug. This is a cell line that was not used to train the model. This is like a new cell line. And as you can see, sometimes could be very good. Sometimes some drugs are very predictable, some are not. So the model not always works. Sometimes works very well. Here is another prediction, different cell line. In this case, it's a breast cancer cell line. The drug in this case is the Citabine. And again, the model sort of gets that right. Now, we'll tell you a little bit more of the model in A3. Now we are going to, so this is what the model is, right, in a nutshell. Now we'll tell you a little bit about another aspect of the model that is more experimentally exploring sensitivities. And here, William Kim is going to describe the same. William?
Yeah, so can you see the mouse? Can you hear me?
We can hear you, but we can't see the mouse.
Okay, well, I'll just describe them. So yes, as Paulo was mentioning, one of the very effective ways to kind of close the loop in this kind of approach, this powerful algorithm, is to then actually test some of these predictions. And, we started out conscious with the understanding that triple negative breast cancer, not only clinically is a highly aggressive disease, but one that's highly heterogeneous. So, we knew going in that there was going to be a lot of complexities. We managed through this project to test highly predicted individual agents, 48 individual agents, that many of which are actually clinically deployed. And we also performed drug combinations, which all sort of accounted for roughly 1,400 or more assays. And we've also deployed some of the models that I describe in the next slide. But one thing that is quite clear, we knew, sort of had expected it going in, but coming out of it, we realized that some of the agents, as a single agents, was not as effective as was claimed in the literature. And that is largely because it's sort of a glass, empty versus a little bit full, and a lot of these cancers are highly resilient to even the most effective agents, some of which have been preclinically tested. So we really kind of switched gears quickly to think about some of the combinations. And so in the next slide. Yeah, so we really kind of wanted to look at combinations that are highly effective, not leaving any cell lines, cancer cells behind, and have found some non-trivial combinations, some of which are highlighted here. So the ones that are in the dark blue really represents this highly effective killing, and each of the columns and rows represent individual drugs, and we see evidence of synergy. Now, synergy has been much rarer than by chance, and this is a result that's been confirmed by others, but certainly this approach allowed us to hone in at least on some of the agents that would be highly synergistic and potentially effective. We also tested this across patient-derived organoids. These organoids were collected through this project in close collaborations with biorepository at the Morse Cancer Center here, as well as Drs. Parker and Dr. Shatsky. And we've collected a number of tumors that then we were successfully able to establish 8 patient-derived organoids. These triple-negative breast cancer organoids are known to be quite challenging to establish. So we're excited not only for the fact that we have those 8 organoids, but then 3 of those are Hispanics. And to my understanding, that those from Hispanic patients are extremely rare. So we have generated some of those. And we profile some of these that were then assessed through the models, some of the models that Pablo was describing. And we were also able to test some of the drug combinations, and some more effective than others. Although in retrospect, some of these or sort of concurrent clinical trials have confirmed that some of these agents are highly toxic in patients. So that's sort of a double-edged sword in some of these drug combinations. So next slide, please. The aim 3 of this really embarked on trying to establish collaborations and really start to deploy some of the samples from from both UCSD as well as the clinic, some of the community collaborators. We, through collaborations with Dr. Shatsky and Dr. Parker, collected 53 unique samples that were profiled, and 14 of these were Hispanics. And we also worked in very close collaboration. Some of the details I'll provide in the next slide of working alongside El Centro Regional Medical Center, a collaboration. And this is a 84% Hispanic community serving hospital, and some of the individuals that really spearheaded this effort are highlighted here. Dr. Parker and Dr. Alfredo Molinillo directed the biorepository, and Dr. Ahmed was the physician at this community hospital. And one of the things that unless you really get into this project, you don't quite anticipate until you, day one, you're starting, you're really trying to establish some of these. And some of the challenges I've highlighted here, others in the later part of the slides. Certainly, things that we didn't anticipate were how there were really lack of infrastructure for research and collaboration, and it's something that we really had to build from ground up, thanks to work of Dr. Parker, sort of the persistent effort, and certainly COVID has really been a big hurdle. And some of the community hospital physicians had an enormous amount of bandwidth issues. It's not to say the academic physicians have less bandwidth issues, but they certainly had some challenges. So I'll highlight some of these in the later part of the slides.
Yeah, so based on having these samples that William described, we can now study, for example, what are the difference between Latina, non-Latina tumors. And here you see a little bit of that on the left. And we can already see a little bit differences in what tumor states tend to be enriching in one particular ethnicity versus another. So these are not all the samples. We still need to add a few. And this will be something we'll report in more detail. We can look at metastatic, another property, basically any phenotypes. Also, we can apply the model to predict responses to these samples from our cohort. So these are not cell lines, these are the actual patient samples. And as you can see, some samples have certain possibilities. There are multiple drugs that are predicted to be potentially useful. And other samples, like the one on the right, seems to be resilient to everything. These are very robust and difficult cancers to treat. And I think what this is telling us is that probably our arsenal of drugs is not complete yet. Another thing that was very interesting is we can study the changes in tumors before and after treatment. And here you can see a transition from one state to another induced by treatment. And these are very interesting. We didn't sort of propose this at the beginning of the project, but I think this is something that we would like to follow up. Now, aim4, William, you want to?
Yeah, so in the interest of time, I think we're running behind. aim4 really highlights some of the activities of trying to engage the community and the stakeholders. This is our pre-COVID CIAPM kickoff meeting with a lot of patient advocates and some of our community collaborators. On the next slide. And some of the reports, this is spearheaded by Dr. Corinne McDaniels, who's with us here today, who put together this report in this very beautiful graphics, and has some very compelling voices being described or heard through this report that again we'll disseminate through the, or we'll share through the report. And I want to highlight, next slide, please, some of the challenges. Certainly, I'm sure everybody really sort of went through this. This COVID really had struck us in a number of different ways. The physical lockdown certainly had effects in how we navigate the physical aspects of the project, but a lot of resignations and a lot of turnovers with the team members as well as the admin staff, both here at our institute as well as the collaborating community institute. So it really sort of highlights the fact that we really kind of, through resilience, persistence, and goal-driven approach, we're able to get to where we are. And so in really sort of the closing, and apologize for the short letters, but we've through this project have put together this really powerful machine learning algorithm that Pablo has mentioned that could be the basis for a lot of guidance towards precision medicine approaches. We've learned tremendous amount of complexities underlying triple negative, some of which we didn't have time to highlight, but we also managed to work with the community from ground up, establishing this collaboration. In fact, this apparently was one of the first and only academic centers in San Diego to establish a remote consenting and through telemedicine, in part due to COVID pandemic. So, hopefully we'll share some of these and more in our progress report. So it certainly takes a village. Here are the individuals that all were, excited to join and be part of this team. And we're really grateful for, obviously, for the generous support of the Precision Medicine California Initiative and the program officers and some of the community physicians, as well as patient. And so Pablo has a next slide. You want to describe the next?
Yes, so sadly, we have to report that our patient advocate passed away recently, a few days ago. Bryce Olson was, somebody that was an inspiration to us. He worked hard against cancer as a patient, and he also was tireless trying to raise awareness of the importance of precision medicine. And one thing I would like to say is that, we face all this challenge, all of us, the complexity of the disease, the problems with healthcare. And so sometimes it's frustrating, right, day by day. But I want you to remember that there are millions of patients that are really rooting for us. Maybe we don't know them personally, maybe they don't know them. We don't know us one by one, but they are there and they are counting on us. So I think we should keep working and know that this is a very meaningful enterprise, despite the challenges and all that. So that's it. I think we're done.
Thank you, Pablo and William, and thank you for that reminder at the end about the people, the patients, the families that you and we are doing this all for. It's not an easy transition for me to ask for questions, but we have time for one question if anyone has one.
Just a quick question. So what type of tissue and information do you need to put as input into this model?
So right now we use DNA and RNA sequencing. We can add other things like protein assays, other types of clinical information. So all that can easily be layered into the model. And that's in terms of the genomics. In terms of the response, anything that provides information about the potential response in the past or in assays or anything, the model will try to correlate that with the genomics and give us a model where, given a new genomic profile, it will find the likelihood of response to whatever agents were used as input. So it's very flexible. As we go deeper into, for example, triple negative, I think certain pathways will be more important and relevant. So the models could be tailored all the way down to a very specific population, or the models could be very general. For example, just predict metastasis in general. So this flexibility is very useful. The challenge is obviously that each of these models require a lot of effort to put in place. But the model is very flexible in principle and powerful from that point of view.
So Elad will talk next, but we have actually a lot of DNA and RNA data that potentially we could put into your model.
Just thinking the same thing, yes.
Yes. Do you have any response data?
Actively being collected.
Yeah.
Okay. Okay. Good.
In progress.
Yeah. So let's talk at some point, and yes, we'll be happy to look at that, and that will be an interesting application.
Yeah. Great.
Thank you.
Thank you, William and Pablo. And those are the exact type of conversations that we hope to foster, today and beyond. With that, I'll introduce our last speaker, Dr. Elad Ziv, who is a professor in the Department of Medicine at UCSF. His research focuses on breast cancer and other age-related diseases with a human population genetics and epidemiology approach, and with a particular focus in Latino and Latina communities, as well as African-American populations. Dr. Ziv received his MD from UCSF and completed his residency and fellowship in internal medicine and clinical epidemiology, also at UCSF. He is joined by members of his project team, including Dr. Susan Newhausen, Dr. Miriam Hernandez, and Alejandra Martinez. And with that, Dr. Ziv, take it away.
Thank you. Let me share my screen. You all see it?
Yes.
Great. Well, thank you for funding us, and thank you also for for inviting us and give us an opportunity to tell you what we have done in the last few years. Sorry. Nope. Something's going wrong. Okay, sorry. There we go. Okay. Try again. All right. So our mission was really to improve disparities, to improve the understanding of disparities in precision medicine. And we broke it down into several components. One is to improve genetic risk assessment, so hereditary breast cancer risk assessment. The second is to improve knowledge about genetic risk assessments among Latinas. And the third is to identify important cancer or somatic mutations in breast tumors among Hispanic Latina women. And I'll just jump right in and tell you, aim one was really kind of the hereditary cancer risk. We broke it down into three sub-aims. One is to analyze variants of uncertain significance in known breast cancer genes. The second sub-aim was to improve polygenic risk scores in breast cancer in Latina women. And the third was to increase knowledge about hereditary breast cancer among Latino populations. So in aim 1A, the goal was to reclassify variants of uncertain significance. These come back more commonly in populations of mixed ancestry or non-European ancestry, just because there's less testing in these populations. And so we started with 4,200 cases and 4,300 controls approximately. This is the results of our first analysis. This is just a high level analysis. These are the odds ratios associated with breast cancer. So if the point here is on the right of the dotted line, it's increasing risk. And if the whiskers or these little bars cross the dotted line, it's not quite significant. And if the color is in the tan color or yellow, those are actually not just a little bit significant, but they're significant, adjusting for all of the known genes in the genome. And you can see for overall breast cancer, BRCA1 and BRCA2, which are obviously the well-known breast cancer susceptibility genes, and PALB2, which is also a well-known susceptibility gene, jumps out. And then we broke it down by ER-positive breast cancer and ER-negative breast cancer. And for ER-positive, the things that were really strong were CHEK2, which also is a very well-known ER-positive breast cancer gene. ATM, which is actually accepted ER positive breast cancer gene was actually not associated in our data, and we're still working out to try and figure that out. And then for ER negative, estrogen receptor negative breast cancer, which is related to, not exactly overlapping the triple negative that you just heard about that the UCSD team was working on, we saw BRCA1 and BRCA2 associated, but also this other gene called FANK-M, which is a member of the Fanconi anemia pathway and had been implicated in ER negative breast cancer before our studies. But to our knowledge, this was the first time it's been identified after adjusting for all known susceptibility genes. And the association that we have found is stronger than has been reported in other populations. That report is a manuscript under review currently. We also managed to recruit 1,679 participants as of, I think, earlier this month. These are Latina women with breast cancer. They recruited City of Hope at UCSF from the clinics at both sites and also from the California Cancer Registry. This was really a tremendous effort by Veronica Tejeda and Anna Fernandez at UCSF, who really did all of the work here. It was a remarkable effort during COVID. We were kind of, unfortunately, having to do a lot of this early on by mail and so on, but they managed to both get started during COVID and work by mail and get saliva samples sent by mail. And then when things reopened, they managed to go back into the clinic and recruit a lot more. So we were very proud of the work that they did. We are now sequencing. We actually completed sequencing, and we're now getting close to done with analysis of 70 genes that are associated with breast cancer, including all the genes that you see on this figure and then a few others, and about 2,500 cases and about 200 controls. And we're going to jointly analyze that with all of us data set in a complete data set that would be about 6,700 cases and over 30,000 controls. And we hope that there will be not only much better variant of uncertain significance classification, but also possibly genes that may be contributing more in this population that have not been well recognized until today. So I would say that AIM-1A gotten pretty much all of we had set out to do, and perhaps a little bit more in the sense that we're beginning to potentially identify new genes that are important in this population. In AIM-1B, the deal was to improve polygenic risk score for breast cancer in Latinas. Currently, we are using genome-wide data from 5,700. So this is array-based data. It's a different technology than the sequencing that I just showed you in the previous slide, although many of the samples overlap. And we have data from 5,700 cases and almost 13,000 controls. The idea for this grant was we were going to analyze what's called the Confluence data set, which would have 24,000 Hispanic Latina women and over 24,000 controls. The Confluence was not going to be, the genotyping wasn't sponsored by this project. It's done at NCI and sponsored by NCI. And it's just taken longer. And so we will be analyzing that at a future date. But actually, Laura Fairman's team has been doing a lot of this analysis. This is the data in terms of the ancestry of the populations, and we've included both women from the US and also women from various studies recruited in Mexico. And what you can see is this is the genetic ancestry. The blue is the European ancestry. The green is the African ancestry. The purple is the East Asian ancestry, which is there. A little bit higher in Peru, kind of as is known, but overall very low. And then the red is the indigenous American ancestry. And that's the two components are really the indigenous and the European. And the indigenous is highest in Peru and higher in Mexico than in the US. And so this is kind of the summary of the work that Laura has been spearheading. And these are polygenic risk scores with various SNP panels that we've included. Sorry, I'm going to have to move the window so I can actually see. But there's the panel on the 3500 SNP panel is sort of the latest European polygenic risk score. And what you can see is that works well in the participants with the lowest indigenous ancestry and consequently highest European ancestry. So if you move to the right, the polygenic risk score is more predictive. But what we had also done before is included SNPs that we had identified. And when you add those back in, what you can see is really a lot better results in all of the populations, but particularly the highest indigenous ancestry populations really move much farther in terms of increased prediction of the model. And so what we're hoping is that with the confluence data coming in, that we'll be able to further improve, particularly in this population, because we'll be able to get better estimates for those rare variants that are specific to the indigenous population. The second part of the work is the educational component that Lara Feherman co-ran with the community partners, Visión y Compromiso and Promotion of Better Health. And what they did was really remarkable work, also having to kind of work around COVID. So they did took promotores who were already working with the population, but needed enough genetics knowledge to be able to become navigators for women who are concerned about their risk, or even just educating women about their risk. And so they did the training over Zoom. They did surveys and actually evaluated how well the educational sessions went. They've already published actually several publications. These are at the bottom. And these are the results. So after the promotores were educated, they then went out into the community virtually initially, and then in person, and did outreach and these training sessions. And the training sessions have reached now over 1,900 women. The mean age was about 44, very low English fluency in this. So the sessions were pretty much all conducted in Spanish. The large fraction of the women did not have health insurance. And actually almost majority had public health insurance, and mostly didn't actually know about genetic testing. They were able to really reach out broadly in Northern and Southern California to do these training sessions, and found that about 7% have very strong family history, who they are now navigating into clinical testing, genetic testing. And this is the team, and also supported in part by UC Davis has a initiative called Lucha, which is really also doing community education around cancer. AIM2 is a tumor mutational profiling aim, and in the first part we were doing our own tumor sequencing, matched germline somatic tumor sequencing to identify variants that actually, genes that are not recognized as, potentially genes that are not recognized as cancer mutated genes, and they're not a part of panels. And then in AIM2B, where we're going to look to see in a much larger data set with Foundation Medicine, if known recurrently somatically mutated genes are more commonly mutated in different ancestry populations. And I'll go talk about that now. So the first part of AIM2A is we analyzed about 146 tumor normal pairs. This is now published. What we found was that this is the somatic mutational profile, so not necessarily looking at any particular gene, whether it's mutated, but basically just the pattern of mutations. And from that, you can often learn about what's driving the tumor. So the most common signature that is in these tumors, and it's also in tumors from other populations, is really a signature that's associated with older age. That's not surprising. Another common pair of signatures is signature 2 and 13. And that's actually a well-recognized signature for something called ApoBec. And there's a common germline ApoBec variant that's actually more common in both Asians and Latinas, and is associated with this signature, although the signature is seen in all populations. So we saw that. We also saw what's called signature 3, which is more commonly seen in the basal subtype of breast cancer, and is associated with defects in homologous recombination DNA repair. So that some of these tumors have BRCA1, BRCA2 mutations and other mutations that abolish their activity to repair double-stranded DNA breaks. And then we also saw this signature 16, which is not something that's been reported in breast cancer in other populations. And Ding, who led this work, actually also saw that a prior publication of a sequencing study in Mexico also saw this signature more commonly than has been reported in non-Hispanic white populations. What was interesting is that this new signature is predominantly seen in the luminal subtype of breast cancer, so not the basal or triple negative breast cancer subtype. And then this is, I would say, very preliminary data that Ding has put together. This is results from 437 women who were sequenced as part of the City of Hope clinical panel. So we had the benefit of working with that as well. And we found that TP53 was the predominant gene. So this is actually TP53 is a well-known gene for all cancers, but it's a little more commonly mutated in the Latina women from City of Hope Clinic than the non-Hispanic white women from City of Hope Clinic. So it's associated more actually with TP53 is much more common in basal or triple negative breast cancer. And so that's something we're analyzing very carefully now. And one other gene that sort of came to our attention is this gene called CTCF. It's a known transcriptional repressor. It's associated with mutations in other tumor types, but not with breast in non-Hispanic white populations. And it is clearly recurrently mutated in this population. Not common, about 3%, but it's clearly playing some role. So this is the analysis now in 40,000 breast tumors with from Foundation Medicine. So Foundation Medicine is a company that's basically sequencing tumors for clinical diagnostic purposes. So they're not doing whole exome sequencing, they're doing more targeted panel sequencing. So that doesn't allow us to look broadly across the genome, but it allows us to look at the known breast cancer drivers and which are more common in a much larger data set. And because they are sequencing everybody, we could look across different ancestries. So we looked at both at African and Asian and Indigenous American ancestry. And these are the results from African ancestry. And what we saw here was that these are copy number aberrations. What we saw is actually just generally copy number aberrations were more common. Copy number amplifications, excuse me, are more common in women with African ancestry. These are both copy number amplifications that's associated with ER-negative breast cancer, which is more common in African ancestry, but also copy number amplifications in red that are associated with ER-positive breast cancer are more common in this population, which is a little surprising. And I think it's underlying the fact that DNA amplifications in general are more common in women with African ancestry. And then we saw PIK3CA is less common in women of African ancestry, but GATA3 is more common. And that's PIK3CA was known before the study. GATA3 is not actually known, some new finding. And then we also looked at the same analysis with Asian ancestry. And there are several genes on the right of the panel that are more common both with Asian and Indigenous American ancestry. One of them I just want to highlight is EGFR. So this is a gene that's not considered a common breast cancer driver, but it was present in a small number of breast tumors, but is also well known as a great area of difference in by ancestry in lung cancer. So EGFR is very common in lung cancer and very common in East Asian populations and to some degree Indigenous American populations with lung cancer, much lower in European ancestry with lung cancer. And what we've identified for the first time is that same pattern holds with breast cancer. So it's overall much less common in all populations, but there's a fourfold difference in lung cancer, and that same fourfold difference exists in breast cancer. And the second panel here, the lollipop plot, just demonstrates that this is true across the gene for all of the known recurrent mutations. And I think this is just sort of our summary slides, and we are working actively to try and maintain the research. Dr. Feherman got a new study to study the genetics of HER2 amplified tumors in Latina women. And Susan and I are working on a grant to try and do more of the sequencing that we mentioned. And there are other grants that are sort of being resubmitted and under review. I think I'll stop there. And I just want to say this was a tremendous team effort by a lot of talented and devoted people, and thank them for that effort. Then I'll stop.
Thanks, Dr. Ziv. We have time for one question for Dr. Ziv. Anyone?
So I have two quick questions. Number one is when you were doing the germline analysis, if you were to divide them up into the young and old, were you going to see some signals become stronger? Because obviously young cancer incidence is in the rise. And then the second question I had was with regard to the somatic analysis that you did. Were these treatment naive samples, or were they sort of post-treated samples?
Right. So I think all of the genetic variants were more common in younger women. We did that, I think, at one point. It didn't sort of jump out at us super strongly. But what I think we saw that was much stronger was that, and I should say, in general, this population is quite young, both the cases and the controls. So, I don't think we have a lot of really old women, but what jumped at us really was more the ER positive, ER negative results. When we finally saw those results, it was almost like, kind of the light had been turned on in the room, and it turns in the sense that, FANC-M really jumped out at us. We went back and read the literature about FANC-M, and actually that's what's always been the case. It's an ER negative or triple negative breast cancer risk factor. We also saw there's a couple of genes I didn't include in there that are good candidates that are potentially novel, and also those came up really much more clearly with the subtype. So I guess based on that, I feel subtype is a little bit more informative than age.
Yeah, we should chat after this meeting, but I mean, I think that even dividing up the groups into ER positive and negative will be able to kind of further subset that into a more sort of homogeneous population.
Yes. The problem is I think we have limited, like we have HER2 on most, but not everyone, because some of these are really historic samples before HER2 is commonly done. And I don't think we have like a lot of other things like, these are all immunohistochemistry kind of, I mean, the subset, so there's about 700 that we have tumor normal on. I showed 146 initial and the 437 that were just more kind of homogeneous from the City of Hope, done in the same batch. But we're putting together a data set with 750, and those are tumor normal. We can do a lot more germline versus like what's going on in the tumor. And to answer your question, I think the second question was, it was tumor normal whole exome. Is that the question?
No, the
Oh,
treatment.
he asked how many were tumor naive or treatment naive.
Oh, treatment. They're all treatment naive. There's a few.
No, that's not true. The
sorry.
136 are all treatment naive, and the
Okay.
37, they're not all. We're still gathering.
Okay.
Information.
Okay. I think I think sorry about that. I meant to show the treatment naive subset. But
you did. That was 146.
Right. But the 400. Okay. Anyway, the final analysis will be, I think, sort of for treatment naive, because I think that it's just harder to interpret the non-treatment naive participants.
Thank you.
So
Go ahead, Isabel.
I was going to say, Elad, can I add? Because I think that while we're looking at this hard science, and I think it's excellent and exciting for our community to have some real look at the issues, I think one of the things that should be pointed out that I was excited about, because Laura and I worked on creating the hereditary breast cancer toolkit that was then tested by Miriam and the promotoras very critically. And one of the things you didn't mention that I thought was important, out of those 1,900 women, 500 were identified as lacking cancer screening, colorectal, breast, and cervical. And this was where we're talking about the early intervention piece, because we know that the smaller percentage are, of course, women with genetic risk. And so that is why we like the combination to everybody of the community engagement piece, because I think we can help identify through the education and awareness the early interventions that need to be done and that are still going unserved, as well as looking at these risk factors so that we can cover the whole thing and get some really impressive numbers and address some of the disparities. I'm super excited about what they were able to learn using, employing our hereditary breast cancer toolkit, and see it as a very valuable added piece for community agencies to be able to start to inform communities at scale about hereditary breast cancer and still identify those patients who need regular screening. Thanks, Elad.
And the other thing, sorry, and maybe Alejandra, the promoters for better health can comment, was, COVID on some level ended up being good because they were able to reach so many more women by doing things through Zoom rather than trying to have in-person meetings or education meetings.
Great, thank you so much.
I just want to echo one other thing for Isabel. Is I think that this is thank you for saying this. I think that disparities is a really complicated thing, and there's disparity. I talked a lot about the disparities at sort of the cutting edge, precision medicine and sequencing and blah blah blah. But a big part of disparities is just like doing like the basic stuff, like that we all know sort of works, and kind of getting that actually done in an effective way, I think is really important, and I'm glad you raised that.
Well, I guess I can just conclude then. I'm pleased to see that all of our projects brought aboard community-based organizations and or community engagement in order to show that across this continuum, the community is really helpful in jumpstarting the work. So thank you very much. I appreciate it. And I loved all the presentations for what I understood for us non-science types.
Dr. Ramos, were you going to say something before I move on? Go ahead.
Yes, thank you. And I think the point was just brought up for us non-science type. So you all are brilliant in terms of really knowing the science and distilling the precision medicine. But I encourage us to now translate that into simple terms for those of us who are not at the bench, for those of us who are not dealing with, and I love Dr. Ziv's explanation of, you know, is it significant, not significant? And so just simplifying it to have others understand, and then not only what the science is showing, but more importantly, the value. Because oftentimes we tend to, and I can tell you, medical speak, we tend to speak in jargon because this is what we know, this is what we do. But I think we need to now simplify it so that others can understand and see the value. And then you're going to have more advocates to say, this is critically important, this is what we need to do. But I was very impressed with the science, and you took me back to my research years in when I had to do all of the tissue culture and all of the basic science. So nice presentations.
Does anyone else have anything to add before I move on? Great. So I just wanted to thank all our speakers, Dr. Patel, who was able to just join us, Drs. Kim and Tamayo, and Dr. Ziv, as well as every member of their project team and community partners, as well as all the participants and patients and families that were engaged in the research. I also wanted to take a minute to acknowledge CIAPM's former program officer and co-director, Dr. Shannon Muir, for her talented oversight of the Cancer Disparities portfolio for almost the whole duration, except for the last few months. I'd like to thank our advisors, Surgeon General Dr. Ramos, Mr. Ron, as well as our Advisory Council Chair, Dr. Lajan Sher, for joining the symposium, and we'll hear remarks from them towards the end of our discussion session today. So that concludes the recorded presentation session for today. So I'll stop the recording and then pass the mic to Julianne to open the discussion session.
Thank you so much, David. And to echo the expression of gratitude, like I mentioned in my opening remarks, it's been just a true privilege to allocate state funds uniquely to cancer disparities research projects with a precision medicine lens. The entire goal of our initiative is to demonstrate the promise and potential of precision medicine, and we really look forward to presenting an evaluation report to the legislature through a process that I'll mention toward the end of our symposium here in the final 40 minutes. But before we conclude,